Now we will study classification of immobilization method for enzyme. There are various methods by which we can immobilize the enzyme. First and foremost is the method for soluble enzymes, and second one is method for insoluble enzyme. Insoluble enzyme involve without derivatization, and another one is with derivatizations. And method for soluble enzymes are entrapment and binding. Entrapment means to entrap something or to entrap enzyme in a fixed space, or we can say in a fixed site. So, type of entrapment include gel entrapment, means to entrap the enzyme in a fixed gel. Second one is microencapsulation, and third one is fiber entrapment. Now, type of binding include carrier binding and cross-linking. Now, carrier binding have the following type: physical absorption, physical adsorption, ionic binding, chelation of metal binding, and covalent binding. Now, we will study one by one what would be the methods for immobilization of enzyme. So, method of immobilization, our first topic is physical adsorption. In this method, our enzyme is being adsorbed on the surface of our substrate. For example, it is the oldest and simplest method of immobilization. In this, enzymes are being adsorbed on the surface. Nelson and Griffin first used charcoal to adsorb invertase for the first time in 1916. The support or carrier used may be of different types. First is mineral support, second is organic support, and third is modified Sepharose and ion exchange resins. Mineral support include aluminium oxide and clay, and the second one is organic support. It includes starch, and the third one is modified Sepharose and ion exchange resin. So, in this physical adsorption, there is no permanent bond formation between carrier and enzyme, only the weak bond. So, the weak bond involved are bonds involved in this are ion interaction, hydrogen bonds, and third one is weak van der Waals forces. Now, there are various methods for physical adsorption. First one is static process, second is dynamic batch process, third one is reactor loading process, and the fourth one is electrode position process. In the static process, we will allow enzyme solution and the carrier solution to come in contact and make a bond, but there is no need of stirring. As it is a static process. So स्टैटिक प्रोसेस वो प्रोसेस होता है जिसमें हम एंजाइम का सॉल्यूशन और कैरियर वाला सॉल्यूशन, द सॉल्यूशन इन विच कैरियर इज़ प्रेजेंट, इन दोनों को हम अलाउ करते हैं टू कम इन कॉन्टैक्ट, बट we don't need to stir the process, we don't need to stir the solution. That is why its name is static process. Now the second one is dynamic batch process. Now in the dynamic process, we will allow the carrier in the enzyme solution and stirring is done. Stirring or continuous agitation. Now in the third process, reactor loading process, carrier is placed in the reactor and then is placed in the enzyme solution and continuous agitation is done. Now in the fourth process, electrode position process, in which carrier is being placed with the electrode in an enzyme bath. Carrier is placed with electrode in the enzyme bath and current is being provided. It will generate electric field resulting in movement of enzyme. Enzyme will immigrates to the carrier and it is being deposited over the carrier. Again, in the electrode position process, we will keep the carrier along with the electrode in an enzyme bath. Then we will provide a current which will generate an electric field. These electric field generation will allow the movement of enzyme towards the carrier, and this enzyme will be adsorbed or deposited on the carrier surface. So advantages of physical adsorption method is no pore diffusion limited. It is easy to carry out, no reagents are required in this process, and it is comparatively cheap method of immobilization. So what are the disadvantages of adsorption method? Desorption of enzyme from the carrier and its efficiency is less. Now the second method is covalent bonding. There is a bonding between chemical group of enzymes and chemical group of carrier. OH and NH2 group of enzyme and carrier can easily form a covalent bond. So alpha COOH group of the enzyme provides important functional group and alpha NH2 group at the end terminal of enzyme. These two are important functional group of enzyme that will provide chemical groups to form the covalent bond with carriers. Now, chemical groups in the carriers that can form covalent bond with the support are amino groups, hydroxyl group, carboxyl group, thiol group, and etc. So, example of carriers or matrix that can be used for covalent bonding are: first is carbohydrates, second is synthetic agent, third is protein carrier, fourth one is amino group bearing carrier, fifth one is inorganic carrier, so example of carbohydrates include cellulose and agarose. Important of synthetic agent is polyacrylamide. Example of protein carrier is collagen and gelatin. Example of amino group bearing carrier is aminobenzyl cellulose, and inorganic carriers include porous glass and silica. Now, methods of covalent bonding include diazotization. Second one is peptide bond. And third one is polyfunctional reagent. In diazotization, there is bonding between amino group of support or carrier and tyrosyl group of enzyme. Whereas in peptide bond, there is bonding between amino group of support and carboxylic group of enzyme. Whereas in polyfunctional reagent, there is a use of bifunctional and multifunctional reagent. There is a bonding of amino group of carrier and amino group of enzyme. Now, advantages of covalent bonding are: there is strong linkage between carrier and enzyme, there is no leakage, and it is comparatively simple process. And now, disadvantages of covalent bonding are: chemical modification of enzyme leading to the loss of functional conformation of enzyme, and sometimes there may be enzyme inactivation by the changes in the conformation when undergo the reaction. Now, the third method of enzyme immobilization is entrapment. In this method, we will entrap our enzyme in a fixed surface area They are physically entrapped in a fixed porous matrix. Bonding involved in this entrapment can be covalent or non-covalent, and the pore size of the matrix can be adjusted to prevent the loss of enzyme. Example of commonly used matrices for entrapment are the first one is polyacryl amide gel, cellulose triacetate, agarose, gelatin, alginates, etc. Now, methods which are used for entrapment are: first, inclusions in the gel; second is inclusion in fiber; and the third one is inclusion in microencapsulation. In the inclusions in gel, enzymes are trapped in the gels, whereas inclusion in fiber, enzymes supported on fiber made of metric materials, and inclusion in microencapsulation, enzymes are entrapped in microencapsules, which are formed by monomer mixtures. Advantages of entrapment is it is the fast method of immobilization, cheap, cost effective, easy to practice at small scales, mild conditions are required. And now the disadvantages of entrapment are: there may be leakage of enzyme, pore diffusion limitations, chances of microbial contaminations, etc. Now, the fourth type of method which is used is cross-linking or copolymerization. Cross-linking, or we can say copolymerization. In this method, enzymes are directly linked with another enzyme of various group by the covalent bond, like various enzymes of various functional groups are bonded with each other. Unlike in this method, there is no use of matrix, no matrix is used. So, advantages of this process is this technique is cheap and simple. It is widely used in commercial preparation and industrial use. Disadvantages may be structurally modify the enzyme, leading to the loss of catalytic property of enzyme. Now, the fifth method will be encapsulation. In this method, the immobilization of enzyme is done by enclosing the enzyme in a membrane capsule. The capsule used in this is made up of semi-permeable membrane like nitrocellulose. Advantages of encapsulation is it is cheap and simple method. In this method, large quantity of enzyme can be immobilized. Disadvantages of this: pore size limitations, and only small substrate molecule is able to cross the membrane.